early 1900s, many scientists used yeast to discover the primary metabolic pathways of fermentation. Yeast lysate was made by breaking open the cells and isolating the soluble contents. Hardin and Young then saw this yeast lysate still produced carbon dioxide and ethanol in the presence of glucose. This then interested more scientists including Gustav Emden, Otto Meyerhof, and Jakob Karl Parnas. These scientists broke apart the components of the solution to discover the EMP glycolytic pathway. This pattern was then found almost everywhere. Glycolysis occurred in a similar pathway for humans, plants, and all bacteria, or so we thought. But then two scientists, Nathan Ettner and Michael Duderoff, weren't able to find these same patterns when looking at Pseudomonas saccharophilus. Ettner and Duderoff then traced back their steps and were able to determine, with some help, this novel metabolic pathway. What Ettner and Duderoff discovered was that their new pathway produced only half as much ATP as the EMP pathway. So our question was, why do organisms still use this pathway when a more efficient EMP pathway exists? This question can begin to be answered through mathematical modeling of these two reaction pathways. From there, they found that the ED pathway releases energy that could be used for ATP as heat, driving the reaction enthalpically, resulting in a higher equilibrium concentration of product. How does this manifest itself in individual reactions in the pathway? The answer is thermodynamic bottlenecks. Bottlenecks are defined as reactions having a free energy change between products and reactants close to zero. Bottlenecks limit the overall flux through a pathway, limiting the rate at which energy can be produced. Flux, by the way, is the number of moles of product produced by a pathway per second. You could think of this as a slide, and children at the top of the slide are reactants and children at the bottom, products. If children go down the slide at a steady rate, they will tend to bump into each other at the flat part of the slide. This is the bottleneck. How can we relieve the bottleneck? The answer is widen the slide. Because bottleneck reactions have only a slightly negative free energy change, the forward flux of the reaction will only be slightly larger than the reverse. In other words, the ratio forward to reverse flux, K plus over K minus, is proportional to the change in free energy of the reaction, delta G, and the equilibrium constant, K. While thermodynamics determines the ratio of flux, the cell can increase net flux by increasing enzyme concentration. For example, if a reaction at a given equilibrium position produces 2 moles per second of product and only 1 mole per second of reactant, one can increase enzyme concentration so that 4 moles per second of product are produced and 2 moles per second are consumed. Same flux ratio and equilibrium constant, but with higher enzyme levels, the pathway can achieve net 2 moles per second of product instead of 1. The cell effectively compensates for a thermodynamic problem using kinetics. In our analogy, we have widened the slide at the bottleneck to let more children through. ED pathway has fewer and less severe thermodynamic bottlenecks than the EMP pathway. So, to maintain the same flux through glycolysis, users of the EMP pathway need to maintain a higher concentration of enzyme. This presents an evolutionary trade-off between efficiency of glycolysis and protein cost. For each individual organism, the answer depends on its environment and the gene set that natural selection has to work with. Mathematical models, however, are still entirely theoretical. To confirm or disprove this model, we need several different future experiments. First, we need to compare enzyme concentrations between similar organisms using either the EMP or ED pathway, and also study the kinetics of individual reactions in both pathways. We could also study under which environmental conditions is the EMP pathway favorable over the ED pathway. Often, pathways reflect adaptation by particular organisms to their unique niche. Overall, we have learned that energy metabolism is not as static as it may initially appear. Mathematical modeling has, without a doubt, provided us crucial insight into the question of why bacteria retain the ED glycolytic pathway over the EMP pathway.